RNA interference is a biological process where RNA molecules are involved in sequence specific suppression of gene expression by double stranded RNA. This mechanism leads to the silencing of gene expression through translational or transcriptional repression. RNA interference has immense potential in suppressing desired genes and is known for its precision, efficiency, stability, and superiority over antisense therapy for gene suppression. The process involves small RNA molecules, microRNA, and small interfering RNA, which play central role in the RNA interference pathway by degrading mRNA and preventing protein translation. RNA interference has therapeutic applications, with the first siRNA based drug receiving FDA approval in 2018, marking a significant advancement in a gene specific therapeutic activities targeting disease related genes.
RNA interference or RNAi is a cellular mechanism that turns off a gene using its DNA sequence. This process is called silencing. A long piece of synthetic double stranded RNA is introduced to the cell, which is designed to be specifically matched up with the target mRNA molecule that encodes for a gene. When a cell identifies double stranded RNA, cellular structures are activated that cut up the double stranded RNA into smaller double stranded pieces, siRNA, or the small interfering RNA. With the help from the guide strand of the siRNA, other cellular structures attach to the smaller pieces of RNA, turning them into single stranded RNA. Now, these single stranded RNA pieces are matched and bind with the mRNA. Once the single stranded RNA matches with the mRNA, it prevents the mRNA from being translated into protein. The matching process stops gene expression from happening, as this normally occurs when mRNA carries the genetic information from DNA to ribosomes for translation into protein.
Talking about the common approaches for RNA interference delivery are lipid mediated transvection and viral mediated transduction. Determining which one of these approaches to use depends upon the cell type using being studied and whether transient or stable lockdown is desired. The most popular application transient transvection of unmodified small interfering RNA or modified stealth RNA. Small interfering duplexes uses cationic lipid based reagent because they are suitable for delivering molecules across a diverse range of commonly used cell lines. For cell type not amenable to lipid mediated transvection, viral vectors are often employed. Adenoviral vectors work well for transient delivery in many cell types. However, for some difficult cell lines such as non-dividing cell and for stable RNA interference expression, lentiviral vectors are best delivery method. Another approach for determining the most favorable RNA interference delivery condition is to use in vitro Zen delivery optimization, scientific resources with extensive knowledge and expertise in viral vectors.
RNAi or RNA interference has a wide range of applications in research. It's used to study gene function, develop new therapies, and even explore potential treatments for diseases like cancer and viral infections. It's like a powerful tool that helps scientists understand how genes work and how they can be targeted for therapeutic purposes. Researchers can use RNAi to selectively silence specific genes and observe the effects, helping them understand gene function and identify potential therapeutic targets. It's also used to study diseases by knocking down disease related genes and observing the resulting changes.
Okay, so let's talk about off-target effects. So similarly, the off-target effects can lead to unintended gene silencing, potentially causing adverse effects. Through assessment and mitigation of the off-target effects are crucial to ensure the safety of RNA-based therapies. RNA interference is a powerful tool for gene silencing, but it can have off-target effects where unintended genes are suppressed. Okay, so these effects can arise due to partial complementary, like between the RNA molecule and unintended mRNA targets, leading to their degradation. So, off-target effects can complicate experimental results and therapeutic applications of RNAi. Such like strategies such as careful design of RNA molecule and validation of target specificity and are employed to mitigate these effects. Moving forward towards safety considerations. Safety considerations are paramount when utilizing RNA interference technology, particularly in therapeutic applications. There are several key safety considerations associated with RNA like delivery, in vivo stability, interaction with drug metabolism pathway, and genotoxicity. So first of all, let me talk you about what is delivery method. The choice of delivery method for RNA molecule is critical for their safety and efficacy. Viral vector, nanoparticles, liposomes, and other delivery systems must be carefully evaluated for the potential immunogenicity, cytotoxicity, and off-target effects as well as. So, another, the in vivo stability. RNA molecule can be susceptible to degradation by nucleases in bodily fluids and tissues. Chemically modification in accumulation and in protective carriers or formulation with stabilizing agents are employed to enhance their stability in vivo. Then, interaction with drug metabolism pathway. RNA molecule may interfere with endogenous RNA processing and metabolism pathway, potentially affecting the pharmacocytosis and pharmacodynamics and co-administrated drugs also. And another one is genotoxicity, where the incorporation of RNA molecule into the genome or interference with a DNA repair mechanism could pose risk of genotoxicity, include chromosomal abnormalities and mutations.